Okay, Assalamu alaikum everyone. I hope you all are doing great. So my name is Lara Samhan, and inshallah today we're going to be talking about the introduction to carbohydrates. So basically before we knew that carbohydrates is as simple as sugars. And basically it is sugars. Before we knew that carbohydrates is just as simple as eating, for example, fast food, which those, for example, bread and everything we eat turns into carbohydrate. For example, like this picture. And then we discovered carbohydrate is way much more like this, and like this. So basically, inshallah, today we're gonna digest each and every single thing in this lecture so that we're gonna understand every point and inshallah simplify it as much as we can. So let's start. Okay. So basically, carbohydrate. What does carbohydrate stands for? Carbs is basically sugar. So how this sugar is basically absorbed into the body? How does it come from, and where does it come from, and how everything happens? Inshallah, we're gonna talk about everything in this lecture. So to start off, as we said, carbohydrates, carbs stands for sugar. So how this sugar comes off? It comes from anything we eat, anything we drink, anything that comes all the way from, for example, the mouth or through IV lines. In general, like anything that comes from outside is actually is sugar. And if it wasn't sugar, for example, like fats, like proteins, it can all the way be digested and absorbed as sugar. Now this thing happened by a couple of processes inside the body, for example, like Krebs cycle, glycolysis, and gluconeogenesis. And we have the store of glucose in the body is called the glycogen store, which is basically a store that is found in the liver. Now, this store is basically a store of sugar that all the way sends out the sugar to the body whenever the body is in need. And for example, let's have an example. For example, a person who ate carbohydrates, sugar, sweets, whatever thing from outside, the body is going to absorb it and store it as carbohydrate or sugar. Now, let's say, for example, a person ate proteins or fats. Even those proteins and fats, it can actually either be absorbed as protein and fats into the body, or it can be transformed to sugar. And for example, it can stay as a store through the glycogen store, and then it can be absorbed in the body as sugars. So basically, sugars in the body can be absorbed from either sugar, protein, or even carbohydrate, from sugar, protein, or even fats. Okay, so basically, we have in this Krebs cycle. This Krebs cycle actually produces for us the sugar or carbohydrates or power in the body throughout couple of processes. I'll not focus more about it in this lecture, but I just get it in I got a couple of slides about Krebs cycle, glycolysis, and gluconeogenesis, so that we know where exactly sugar comes from, how does it happen, and to have a clear picture of everything, how it happens. So basically, in this Krebs cycle, the only thing I want you to notice is that, and the most important thing about this Krebs cycle is that it produces for us power in the form of FADH2, NADH, GTP, and those productions, this and this actually is basically like ATP, which gives us the power. Tamam. This is the first thing in regard to Krebs cycle. We also have the process of glycolysis and gluconeogenesis. Gluconeogenesis, as the word says, gluco and U. U means something in you. So basically, from fat and from protein, it all the way throughout this process, it turns out to be sugar. And the body uses the process of gluconeogenesis. Whenever we do not have enough sugar in the body, it all the way takes the store of the protein and fat and transform it into sugars. So that the body is gonna have power and have more energy. This is in regard to gluconeogenesis. And also we have in the process of glycolysis, it starts off with glucose and ends up with pyruvate. And the most important thing in those three cycles is that we need power, we need energy, and we need ATP. Okay, so moving on. Okay, also this slide basically explains how we use sugar, or in other words, the process of using sugar. For example, for whenever you eat sugar, you can absorb it and you can actually use it in the body. If, for example, for whatsoever reason, we didn't have sufficient for example, eating of sugar, so the body is gonna use the store, or in other words, the glycogen store, which is in the liver that has sugar, and then this sugar will be all the way sent out of the liver by a couple of processes as sugar. And this glycogen store is used, for example, the x-axis is basically the days and the hours. So this glycogen store can be used from this time, let's say from 4 or 5 hours, all the way to 1 day, which is 24 hours. The body can use the glycogen store that has the sugar, And the gluconeogenesis, as we said, the new is proteins and fats that's all the way transformed to sugar. This story can actually start off from a couple of hours all the way to days, which can actually reach for more than 1 month or approximately more than 40 days. Okay, so this is just a quick review of like how our body uses the sugar and from where it comes. Now, coming up to our objectives of this lecture, we want to know more about the carbohydrate, the function of carbohydrates, different type of carbohydrate, the monosaccharide, disaccharide, polysaccharide, and the oligosaccharide. We want to know more about the structures. We want to know more about the glucose transporters or the GLUTs. And okay, so let's get started. Okay, so now what is carbohydrate? As we took in basic science in high school, carbohydrate formula is actually this formula, which is CN. N is actually any number, 1, 2, 3, 4, 5, or any number, CN, H2ON, and this is like the basic formula of carbohydrate. Now, carbohydrate, as the word says, is sugar, and as we know, sugar gives us energy, gives us power. Now, in regard to the functions of carbohydrate, it gives us energy. We have the energy store. As we said, the glycogen is the store of glucose, which gives us energy. We have the glucose isn't only actually used to give us energy, but it's also used as cell wall. It's also used to help in the transmission, and it basically has a lot of functions. So it's available in the cell wall and the exoskeleton. It helps in the signaling, cell-to-cell signaling. And if the glucose is attached to the protein, it has two names, either glycoprotein or proteoglycan. Now let's understand what does glycoprotein means. Glyco is glucose, or in other words, sugar. Protein is a protein. So like glucose with protein. And we have the other word, which is almost the same, proteoglycan. Proteo means protein, glycan is the glucose. So protein with glucose, and here glycoprotein means glucose with protein. They almost have the same exact meaning, but with many differences, which we'll get to know more in this lecture. Okay, so what does types of carbohydrate mean? As we said, we have mono. Mono means one, which is one molecule of sugar. Di means two. We have two molecules of sugar. Oligo means a few, which is few molecules of sugar, or 2 to 10 molecules of sugar. Poly means many. We have a lot of molecules of sugars. So those are the different types of sugars. Now, in regards to this slide, basically just explains how the glucose molecules help in the signaling. So basically, let's say this is the cell, this is the whole cell, and this is the cell wall. As we said, the glucose can be also found in the cell wall. For example, we have this thing. This thing is basically a protein, and into this protein we have on the above, this thing is basically a sugar. This is a sugar. This is a sugar molecule. So basically, it just tells us how the sugar is helping us in the transmission, cell-to-cell transmission. For example, we have here from the outside, this is the receptor of whatsoever molecule. Either it is, for example, a virus, like influenza virus, or a bacteria, or for example, a neutrophil, or let's say for whatever thing, or a lysosome, whatever thing those things are from outside, it has a receptor. Now, this receptor want to, for example, bind with this cell through the receptor of this cell. Now, this receptor or this protein thing basically has a sugar, and this sugar molecule helps in the efficient binding of both cells. This cell, for whatever it was, bacteria, virus, whatever thing, and this cell. So this is like one of the functions of this sugar, and it just says that this sugar is basically an oligosaccharide. It can be either monosaccharide, di, poly, or even oligosaccharide. In this case, this sugar is basically a oligosaccharide. So this is the most important thing in this slide, just receptor to receptor binding by the help of this sugar molecule. And then we're going to have, after both of them bind together, we're going to have the signal transmission throughout the cell. Tamam? Okay, so moving on. Now let's get to know more about the carbohydrates. As we said, carbohydrates is basically sugar. It can be different type of sugar. We have polysaccharide, disaccharide, mono, and oligosaccharide, as we said. So in regard to monosaccharide, we said mono means one, which is only one sugar. Oligo is few, and so on. So we said that the glucose, basically we use for the carbohydrate, it has a formula, as we said, and in regard to the glucose, it has 6 molecules of sugar. Because of that, it's C6. So the sugar formula, as we know from basic high school, is C6H12O6, and this is actually the most common monosaccharide. Now, in regard to the monosaccharide, it can be either classified by the location of the carbonyl group or by the number of carbons. Now, what is carbonyl group? Carbonyl group is basically, as we can see in this figure, it's a C molecule or a C atom attached with double bonds with O, which is actually the oxygen. This is the carbonyl group. Now, if this carbonyl group, which is the C double bond O, is attached to R, which is in other words carbon, R stands for carbon, is considered a ketone. If this C double bond O is attached to H, atom is considered as the aldehyde. So we have two things. If the carbonyl group is attached to H, it is aldehyde. If the carbonyl group is attached to R, which is in other words C or carbon, it's called as the ketone. Now, why this carbonyl group is important? Because when we draw the carbohydrate molecules, the different type of carbohydrate molecules, it's basically important to know from where we are going to count, for example, 1, 2, 3, 4, and it helps us, for example, to know how the structure looks like. It helps us to know, for example, is it the mirror imaging and everything. Like this has a really important role in the of how the carbohydrate molecule looks like. Okay, so moving on. Okay, so now as we said, we have the aldehyde and the ketone, or in other words, the aldose and the ketones. Now, once you see any word that ends with OSE, immediately this is a sugar. Whatsoever word in the whole world that ends with OSE, خلاص, this is immediately a sugar. For example, we have aldose. For example, let's assume you don't know what aldose is. Once you see OSE, خلاص, you know at least it's sugar. OSE, خلاص, you know it's a sugar. So for example, we have in here this sugar molecule, which is aldose, contains aldehyde, like from the words aldose, aldehyde, ketose, ketone. As we said, aldehyde, for example, this is an aldose. It has a carbonyl group with H, so this is a aldehyde. It has, for example, C double bond O with C molecule, for example, from this side or even from this side. That means this is a ketone. In this case, we have ketotriose. Why ketotriose? Tri means 3. And ketone means the carbonyl group is attached to a C molecule. And here the C molecule, the carbonyl group is attached to H molecule. Because of that, it's aldose. Now we have tri in here and tri in here because we have 3 carbon molecules in this side and 3 carbon molecules in this side. So we call it as aldotriose in this case and call it as ketotriose in this case. Other word for this molecule is basically glyceraldehyde, and here we have the dihydroxy acetone. Okay, so di means 2, hydroxy. We have over here OH. This is the first one, and this is the second OH. So dihydroxy acetone. Okay, perfect. Now we have in here just any word that ends with OSE, خلاص, you know that it's immediately sugar. So we have triose, OSE, and sugar, 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 sugar. So basically, those are different type of sugars. Tri means 3, so a sugar molecule with 3 carbons. Tetra is 4, pent is 5, hex is 6. So basically, we have in here 6 carbon molecules, carbon atoms with the that is basically sugar or carbohydrate. Now, in regard to the ribose, it just says that it's basically 5 carbon sugar. Glucose is the most common type of monosaccharide, which is a 6 carbon sugar, and fructose is basically a ketone, and it's a form of glucose that is a 6 carbon atoms. Okay, so moving on. So now in this slide, we're just focus more into it because let's get more oriented. So basically, we have in here the aldose and the ketones. Type. We said aldose is basically C double bond O with H, and ketose is C double bond O with a C carbon. Tamam. Now we have in regard to the aldose, as we can see, we said it's C double bond O with H. As we can see, this example, this example, this example, and this example. This is in regard to the aldose. Same goes to the ketose. We said it's C double bond with C. Okay, so we have a question. Just 1 minute. Let's see it. Okay, sure. So we're going to explain the difference between the aldose and the ketose. Aldose is basically, both of them, the aldose and the ketose, has C double bond O, as we can see in this picture, and as we can see in this picture. Now this is clear. The only difference between them is that the ketose, or the ketone in general, it has C double bond O, is attached to a C carbon atom, and it's attached from either side, either this side or this side. It's attached also here to C carbon atom. And for the aldose, the C should be attached at least from one side to an H atom. Here it's attached to a C atom, صح. Like in this side, if it's only attached to one hydrogen molecule, one hydrogen atom, خلاص, this is considered a aldose. So we have C double bond O With H, then it's aldose. Here we have C double bond O with C, either from this side or this side, خلاص it's ketone. And usually in regard to the aldose, the H is usually attached at the first carbon molecule. Usually, خلاص once you see the first carbon molecule is attached to H, you're gonna say خلاص it's aldose or it's aldehyde. Usually the ketone, the C double bond O, is attached to C molecule, C atom. So basically, it's gonna be found at the middle. So this is a ketone. So as simple as that. C double bond O with H, aldose. C double bond with C, this is a ketone. Is it clear? Okay, perfect. Okay, so this is the difference between the aldehyde and the ketone, or the aldose and the ketone. خلاص, now we know everything in here. We agree this molecule, those two sugar molecules are aldehyde or aldose, because we have C double bond O with H. Here, everywhere. And for the ketone or the ketose, we have C double bond O with C. Here in the middle, here and here. So خلاص, we agree that this is ketone. We agree that this is aldose or aldehyde. ممتاز. Now, for example, we have triose, pentose, hexose. As we know, O's is sugar. So we have tri sugar. Pent is 5 sugar. Hex is 6 sugars. As we can see here, 3 sugars. First carbon molecule, second carbon molecule, third carbon molecule. خلاص, this is triose. We can see in this thing how many carbon molecules? 1, 2, 3, 4, 5. So 5 carbon molecules is pentose. 6 carbon molecules, 1, 2, 3, 4, 5, 6 is hexose. Same goes in here. 6 carbon molecules is hexose, 5 is pentose, and 3 carbon molecules is triose. Now, each molecule has different shapes or has different figure into the space. So, for example, we can name this thing as triose. We can name this thing as triose. As we can see here, the C double bond O with H is aldehyde, but like in here, it's ketone. So they have different names, even though we have here 3 carbons and here we have 3 carbons. So this is called as glyceraldehyde. As we know, glycer is glucose aldehyde because we have a aldehyde group. This is ribose, which has 5 sugars. This is called as ribose. This is called as glucose, and this is called as the galactose. The only difference between glucose and galactose, even though both are 6 carbon molecules, is that here in carbon number 1, 2, 3, 4, and carbon number 4, the OH and the H are basically the opposite. Exactly, ممتاز. Okay, so same goes all the way to the fructose, ribulose, and the dihydroxyacetone. It's basically acetone because it's something related to the ketone. So dihydroxy because we have 2 hydroxy groups, as we said. Here we have the ribulose. Anything that ends with O's is basically a sugar. Fructose, anything that ends with O's is basically a sugar. So خلاص, now, alhamdulillah, we are oriented to how the figure are. We understood how they are. We understood what's aldehyde. We understood what's ketone. We understood what's OSE. Anything that ends with O's is basically a sugar. So alhamdulillah, now we at least understand what are basically those things. Now we have in this right-sided figure, it just explains more about the triose, tetrose, pentose, hexose, and the heptose. Hept is basically 7, so we have 7 carbon molecules. And here it just gives examples of the hexose, of the heptose, example of the hexose, and as we know, glucose is the most common type of monosaccharide or the hexose. And here it just explains, for example, galactose is basically found in the milk. Same goes to the glucose. It's the major energy that we have, and it's the most common type of monosaccharide. Same goes to the pentose, different type of pentose, tetrose, and the triose is basically 3 carbon molecules. So this just explains each and every type of sugar and what type it is. It's almost the same as what they explained. Okay, so is everything clear so far? Perfect. Tamam. So now let's get to know more about this thing. Okay, so this slide is really quite important, so I'll spend quite enough time in it. Type. Now let's start off with the isomers. This is the isomer. Isomers are divided into 2 things, the constitutional isomers and the stereoisomers. Stereoisomers is also subdivided into 2 things, which are the enantiomers or the enantiomers and the diastereomers. Diastereomers is basically subdivided into 2 more things, which are the epimers and the anomers. Yeah, let's get to divide one by one. We start off with the isomers. What are isomers? They are basically the exact same formula or the exact same molecular formula with different structures. They have different structures in the space, either this constitutional isomers or the stereoisomers. So basically, let's have an example. We have in here, we have C3H6O3. Same exact thing in here. C3H6O3. They have the exact same number of molecules. They have the same molecular formula, لكن their basically figures into the space is different. This one is different from this one. طيب. So let's get to know more. This is the meaning of isomers. Now خلاص we know what is isomer. Same molecular formula with different structures into the space. طيب, we have two types. We have, as we said, constitutional isomer and the stereoisomer. Now, what is constitutional isomers? They basically differ in the order of attachment of the atoms. So they have the exact same formula with different structures. As we said, aldehyde, here the name is glyceraldehyde. Aldehyde because it has, it's aldehyde like aldolose. So we have C double bond O with H. This is glyceraldehyde. Glycer is glucose. This is dihydroxyacetone. Acetone is more towards ketone. So we have C double bond O with C in this side and C on this side. And it's dihydroxy because we have two hydroxyl group. This is the first and this is the second. So this is why we name it as this thing. So alhamdulillah, now we are good with names, at least, even though if we forget the names, we can invent some name which is like, inshallah, most probably correct. طيب, ممتاز. Now we have in here, this is the exact molecular formula, ولكن they are different in the structure from both sides because of that, we have different names. تمام? This is called a constitutional isomer. So for example, doctors usually, like, the most common question that they get is that they get two figures like this and like this, and they're gonna ask you, is this, for example, constitutional isomer, ولا is it anomer, ولا is it epimer? So خلاص, you know, they have the exact same formula ولكن different structures. على طول you will choose it's a constitutional isomer. This is like usually how questions come. So this is a constitutional isomer. Now, alhamdulillah, we are good with constitutional isomer. For the formula, I'm not sure صراحة. Maybe they will give it, maybe not. Depends يعني on the doctor's mood. بس I'm not sure صراحة. Maybe they will give it, maybe not. يعني I can't give a word صراحة. Exactly. If basically the doctor didn't give a formula, you just count the number of carbons. You know that the formula of, the formula, for example, of the carbohydrate starts with C and then H and then O. So you count how many number of C, how many number of H, and how many number of oxygen. But inshallah, let's hope that they get the formulas. تمام. But if they didn't get, خلاص, you just write CHO and count how many numbers of carbon, how many numbers of H, and how many numbers of oxygen molecule. Okay, perfect. So this is called as the constitution. Now, alhamdulillah, it's crystal clear. Now let's get to know what is a stereoisomer, which is the second type of the isomers. Now, it's basically atoms that are connected in the same order, but they are different in the spatial arrangement. So they are connected exactly, for example, in this case, exactly in the same order, ولكن they are different. ولكن here they are also different. How they are different? For example, let's take this example. مثلا here we have CHO. ممتاز. C carbon, excellent. Here we have the H is on the left side, ولكن here the OH or the hydroxyl group is on the left side. So we don't have here H on the left side, the same as this thing. So if they are different in this thing, خلاص, we're gonna call it as stereoisomers. Okay, ممتاز. طيب, now let's get to know the different types. Now we know what is stereoisomers. They are basically atoms that are connected in the same order, ولكن different in space. So we have two types. We have the first type is the enantiomers. The second type is the, okay, I'm going to explain it again. So we said in regard to the isomers, we have two things. The constitutional isomer, they have the same formula, ولكن they look different. This is different than this, خلاص, constitutional isomer. We are clear with this. The second type is stereoisomers. It's basically atoms that are connected in the same order. For example, you have here how many C carbons? 1, 2, 3, 4, 5, 6. Also here you have 6 carbons. طيب, here we have how many carbons? 1, 2, 3. Also here we have 3 carbons. So they assume to be looking the same. Like when you have the first glance on this structure and this structure, you will say, okay, they look almost the same. Same goes to this structure and this structure. Once you look at them, you will say, oh, okay, they are the same. صح? Like in regard to the carbon molecules and in regard to their arrangement, in this case, they look the same. Same goes to here, they look the same. Same goes to this structure and this structure, they look the same. Like the first glance, you see that they look the same. Same goes to this structure and this structure. The first glance, they look the same. Do we agree about this thing? ممتاز, روعة. خلاص, this is the stereoisomer. It means that they exactly look the same, same. All of them are exactly looking the same, and they exactly have the same formula. مثلا هنا C3H6O3, كمان هنا C3H6O3. Same goes to this formula and this formula and everywhere. So they look the same in structure, they assume to look the same, but they are differently oriented into the space or in the spatial arrangement. هنا وهنا. Same goes here, same goes same process. تمام, we have a question. Exactly. Now I'll explain everything. Don't worry. So we agree that, let's repeat everything again. Isomers, they basically have the same molecular formula but different structures. We have two types: constitutional, stereoisomers. Constitutional, they have same formula ولكن they look different. The structures look different. Once we see different structures, خلاص إن شاء الله this the answer is constitutional isomer. أما بالنسبة للstereoisomers, they have the exact same formula and they assume or they look same in the spatial arrangement ولكن we have some differences. تمام. So once we see they look the same, they look the same, they look the same, they look the same, خلاص, the first glance they look the same, and this is a stereoisomers, and we're gonna remove from our differentials that this is constitutional isomer. خلاص, now we agree all of the structures are stereoisomers. Clear? Amazing. Now we have the first type of the stereoisomers is the enantiomers. The second type is distereoisomers. Now I'll talk more about the enantiomers. As we said, we have the exact chemical formula, C3H6O3. Same goes into this thing. Now we have here this is called, or this molecule is basically this sugar molecule is called the glyceraldehyde. What does D mean? D means that the OH is basically on the right side. So once we see the OH is on the right side, is basically D. Here it says L. L means left. Just for the sake of memorizing it, just know L is left. So the OH is basically on the left-hand side. This is the OH on the left. So once the OH is on the left, it's L, and then we can write the name of the glucose molecule. And once, or the carbohydrate molecule, and once it's called as D, that means the OH is on the right side, that means it's D. So now L and D is clear. ممتاز جدا, excellent. طيب, now we have this is glyceraldehyde. We have aldehyde. Why? Because we have the C double bond O with H. So this is an aldehyde group. So it's a glycer. It's meaning يعني carbohydrate or glucose aldehyde. So D glyceraldehyde. Clear. This is L because the OH is on the left side. Glycer is like glucose, and aldehyde because also we have an aldehyde group. C double bond OH. ممتاز. So now we know the chemical formula. We know why it's called as D, why it's called as L, and we know exactly what's glyceraldehyde, and everything is clear, alhamdulillah. Now we have over here a chiral carbon. Chiral carbon is basically a C carbon that's attached to 4 different things. This is the chiral carbon, and it's usually after the C double bond OH. But the most important thing about the chiral carbon is that a C attached to 4 different molecules of atoms. This is known as the chiral carbon. This is the chiral carbon. Okay, so this is the meaning of the chiral carbon. Now, what does mirror image mean? Mirror image, that means if I put a mirror into here, they will exactly look the same side. For example, like an easy way to just visualize it instead of putting a mirror and like how does it look like? Just say over here the OH is beside the OH. So this is like if they are beside each other, this is the first mirror image. Second thing, you just move like atom by atom, so that you know if they are mirror, like so that you're going to be sure that both are mirror images. مثلا هنا يلا OH with OH, إذا if they are close to one another معناها هاي mirror image. يلا الحين C double bond H with C double bond H. So it's a mirror image. The H outside with this H from the outside. So both of them outside and outside are mirror images. This is down CH2OH. This is down CH2OH. So both of them are down. Is a ممتاز. It's mirror image. Now we understand what's mirror image. Clear? ممتاز. This is a mirror image. Now, what does it mean non-superimposable? It means that we can see there is difference between this thing and this thing. معناها if, for example, I put 2 molecules onto each other, no matter how much I put them onto each other, they will never be exactly the same. Why? For example, here the OH is on the left side. ولكن here there is no OH on the left side. Because of that, no matter how much I put them on top of each other and just orient them around, they will never be on top of each other. If you just imagine that you're folding both of them on top of each other, they will never exactly be the same thing. Why? Because they are non-superimposable. So non-superimposable, it just means that both molecules are different because this OH is on the left side, and compared to this H is on the left side, but there is no OH in here. So because of that, once you see there is difference in the orientation of the molecules, that means it's non-superimposable. So let's repeat again. Mirror images, that means if you put a mirror in here, OH beside each other, this is beside each other, H and H are far, and this is also far. This is CH2OH and this is CH2OH. So alhamdulillah, they are mirror images. ولكن if I put this one, just copy paste this one all the way on top of this one, you will see that this side there's H and this side there's OH. So because of that, they are called non-superimposable. Now, non-superimposable, usually people confuse it, but I want to make sure you understand it. It can be basically anything. Inshallah, can aldehyde, can be ketone, as long as you understand the idea that they have the same formula. They actually almost look the same, but there is many differences. Whatever was the formula, inshallah, whatever it was, خلاص, as long as you know the concept, it applies to like anything. Clear? Okay, ممتاز. So now we understood the enantiomers are basically having the same chemical formula. They assume to be having the same, to look the same into the space. ولكن they are basically mirror images, and they are non-superimposable. That means if we put this one on top of this one, they will never be exactly the same, no matter how much we just moved it around. This is enantiomer. خلاص, alhamdulillah, enantiomer now is clear. Let's move to the diastereomers. It's basically isomers that are not mirror images. طيب, we said mirror images. Let's assume there's a mirror in here, and let's see. Let's move from top to bottom. CHO, CHO, mirror image. H, mirror image. OH, OH, mirror image. OH and H, are they the same? No. That means this is not mirror image. Type OH and H. Also, this thing is not a mirror image. So we have two things that are not mirror images. Because of that, it's called as di. Di means two. So we have two things in this structure that are not mirror images. So let's repeat again. What are diastereomers? We have in two positions of the carbohydrate molecule two things that are not mirror images. In this case, we can see this first thing, OH and H is not mirror image, and we have the OH and the H is not mirror image. This is mirror image, yes. This is mirror image, yes. CHO, CHO mirror image, yes. CH2OH with CH2OH mirror image, yes. So alhamdulillah, the whole molecule is mirror image, except this thing and this thing. So we have two things. So because of that, it's called di, or in other words, diastereomers. Di because two, stereomers because stereoisomers because it's under the stereoisomer type. Okay, so they have the exact same chemical formula. They look to be same into the space. Our spatial arrangement is almost the same. ولكن we have two things that are not mirror images. So once we have two things not mirror images, immediately خلاص, this is diastereomers. Clear? Thus, now this diastereomers, we have two types of it. We have the first type is the epimers, and the second type is the anomers. Anomers is very easy. Once you see a structure that is a ring-like structure, without even checking anything, خلاص, you can say, if this was the only choice that was a structure looking like, it is immediately anomers. So what's an anomer? It's basically isomers that are that differs basically, that it is basically an asymmetric carbon formed by a ring. Let's look into this thing in here. For example, this is a ring-like structure, and we have, it's basically called a pyranose ring, or in other words, it has a 6 carbon molecule. How to count the carbon molecules? We have here number 1, number 2, 3, 4, 5, and 6. So 6 carbon molecule is basically a pyranose. Os means sugar. Pyranose as a word, pure basically is like a 6 carbon molecule. So a sugar of 6 carbon molecule, and it's a ring-like structure. Anomeric carbon. Now, what does the word anomeric mean? For example, this ring was actually like this. It was like, for example, as a Fisher arrangement. Fisher arrangement is basically like looking like a ring, and looking like a for example, C, like in this arrangement is called Fisher arrangement. Now, if I want to change the Fisher arrangement into this thing, into a ring-like structure, so basically I fold it all around, and it will look like this thing. Tamam. So once this, once it's all the way close together, and we have this C-level bond O comes in here. So basically, this thing is called the anomeric carbon. I'll just repeat it again. So we have this thing is basically not a ring-like structure. It's just a Fisher structure. Once I close it, if I want to close it and form it as a ring-like structure, it will look like this. Now we have the C-level bond OH, for example, like this thing, C-level bond OH. This will actually come in here, C-level bond OH. Tamam? So this carbon that's basically closing the ring, ending the ring, is called as the anomeric carbon. So in here is the anomeric carbon, and in here, like the ring closes at this point. And usually it's very common for the ring to close at this point, like in case you were confused. Usually it's here. Sometimes it differs rarely, but yeah, most of the times it's usually here. Tamam? So this is the anomeric carbon, this is the anomeric carbon. ممتاز. Now, we said anomers is basically just a ring-like structure. Once you see a ring-like structure, خلاص, immediately this is anomers. We have in here, as we said, they have the exact chemical formula, C6H12O6. Same goes into this thing. Okay. Okay, I'll explain how it's C1. Usually the anomeric carbon is carbon number 1 because it's the thing that we close the ring into it. For example, like this thing, we have over here CHO till the end. Tamam, tamam. Now, for example, same goes, okay, so we have this thing in here. Now, if I want to count this thing, this is carbon number 1, 2, 3, 4, 5, 6. If I want to close the ring, this will be carbon number 1 because it's the place where the first carbon is. Tamam? Because of that, we call it as carbon number 1 or anomeric carbon. Clear? We're gonna have more examples on it, don't worry. Tamam, perfect. So this is carbon number 1, this is carbon number 1. Mumtaz. So we know what is anomers. Now let's get to know, and we know the chemical formula. Now let's get to know how it's named like this. This is basically a sugar or a pyranose ring. Mumtaz. We have in here D. We said D is basically it's on the right side. That means that the OH is basically on the right side. This is D. And same goes into here. D is on the right side. So basically in this structure, we don't know where's the right, where's the left because it's a ring-like structure. Lakin if it was open, we will know that the OH was on the right side. Same goes into this thing. Tamam, tamam. D means right side. The OH is on the right side. L means the OH is on the left side. Mumtaz. Now we have in here something new like alpha and beta. What's alpha? Alpha is basically opposite. Just remember, alpha looks like an O, so it's opposite. Beta is basically same side. What, like opposite to what and same side to what? We have in here the OH group. Just look at the anomeric carbon, which is this one we said in the structure, and it's also this one, anomeric carbon. We always compare to know alpha and beta, the OH carbon, this thing, into the CH2OH. We have in here the OH carbon with the CH2OH. Perfect. Now, if this OH is basically looking down, right? So we can see this is the ring. The OH is pointing all the way downward. Tamam, tamam. And when compared to this OH, which is all the way pointing upward, صح ولا لا? So we will compare the OH to the CH2OH. Same goes in here. We're going to compare the OH to the CH2OH. ممتاز. Now, if this OH is pointing down, we all agree, and this OH is pointing up, we all agree. ممتاز. So this OH is pointing down, and this CH2OH is pointing up. So this is pointing down, this is pointing up. So it's opposite to one another. Because of that, we call it as alpha. Compared to this thing, the OH is going up, and the CH2OH is also going. Both are going up. That means it's beta. Now, is the alpha and beta concept clear? ولا مو clear? Amazing. ممتاز جدا. So I'll just review it على طول. We have alpha is opposite side, beta is same side. What's opposite and what's same side? If the OH group in the anomeric carbon, which is carbon number 1, down, and this is up. If they are opposite sides, it's alpha. Remember, alpha is like this. Alpha is O, يعني alpha O. So this is opposite side. O is opposite. ولكن if the OH is up and the CH2OH is also up, so both of them are pointing to the same side, معناها it's beta. So alhamdulillah, now we know the chemical formula, we know the structure, and we know why it's named like this. So whatever molecule comes in the world to you, you already know how to name it, even though if it was your very first time to see this molecule. ممتاز. So this anomer is basically a type of the diastereomer. Now let's come to the second type of the diastereomer, which is basically the epimer. Epimer is basically differ at one of several asymmetric carbon. Epi means one. So epi means one. One معناها it's only differ into one carbon. How it differs? Let's see. For example, here the chemical formula is exactly the same. C6H12O6. Same goes into here. خلصنا. يلا, now we have this D. It means the OH is on the right side. Same goes in here. The OH on the right side. خلاص, now this is clear. Glucose and the mannose. Anything that ends with O's is basically a sugar. Anything that ends with O's is a sugar. يلا, now we said epimer. Epi means one. معناها it's different and only in one carbon. Let's see. مثلا هنا CHO. It's the same. هنا عندي C. هنا عندي C. لكن هنا the OH is on the right side. ولكن here on the right side we have the H, right? So they only differ at one carbon. طب let's continue. مثلا here we have C. Here we have C. Here we have H on the right side, H on the right side, it's the same. Here we have OH, here we have OH, both of them are on the right side, ممتاز. Here we have OH, here we have OH, both of them are also on the right side. And here we have CHO. Is a ممتاز, isn't the whole structure or the whole Fischer structure is exactly the same except only in one carbon. Here the OH is on the right side, أما هنا the H is on the right side. If it's only different only in one carbon, معناها it's epi. Epi يعني one, it's called as the epimer. Is it clear ولا مو clear? That's it then. So I'll just do a very quick review because like this is the most important slide in this whole lecture. And usually questions come from this thing. They get you basically a structure. Like mostly in previous batches, they usually get questions from this slide. For example, they get you this structure. They tell you this like two structures. They tell you is it a constitutional isomer, or is it diastereomer, or like this. So because of that, I focus mostly on this slide. Okay, so we have a question. Never look at the bond, just look at the carbons. Like the carbons are the most important thing because usually the different type of carbohydrates have different names based on the carbon. How many carbons are there? Is it 1 carbon, 2, 3, 4, 5 carbon? So like the carbons are the most important thing to count. But generally speaking, just look at the structure itself and just know the difference between the isomers, stereoisomers, constitutional, till the end. If you know this thing, خلاص. Usually the usual question that comes from the carbohydrate is that they get your structure and tell you what type is it, or like this. Like this is like the most common thing that comes, even though other thing can come, but yeah, mostly like this is like the most important concept in this lecture. So I'll do a very quick review. Yalla, now we have the isomers. Isomer is basically same molecular formula ولكن different structure. We have two types, constitutional isomer and the stereoisomers. خلاص, constitutional isomers is basically they have the same formula ولكن they look different. Once you see two different things, case closed. It's constitutional isomer. Now this is now in regard to the stereoisomer, that we have a lot of things into it. It has two types. The first is the enantiomer. The second is the diastereoisomers. So the stereoisomers is basically into two things, the epimers and the anomers. Once we see a ring-like structure, خلاص, this is ended as a anomer. خلاص, we ended. Once we see epimer, only one carbon atom is basically changed in one one carbon atom difference between this structure and this structure. For example, here the OH is on the right, here the H is on the right, so they are only different in one carbon. خلاص, it's epimer. And diastereoisomers, once we see they are basically different in two things. For example, here and here, and خلاص, this is diastereoisomer. Or for example, here, for example, the OH is on the right, OH on the right, OH on the right, same goes in here. ولكن, for example, they are different in two things. This thing, here it's H, here it's OH, here it's OH, and here it's H. If they are only different in two carbons, معناها it's diastereoisomers. You can actually have a couple of ways to look at either to look at it into here, the H is on the right side, OH is on the right side. Same goes in here. And now we have two things. Or you can look at into it like this. For example, here the OH and here there's H. Like it's not mirror images. So basically, you can look into it in different ways as long as you get the answer correct. That here the difference is only in two carbon difference. عشان كده it's called diastereoisomers. تمام. Compared to the enantiomers is basically they are non-superimposable mirror images. Mirror images, this is same as this. This is same as this. Here the OH is mirror image to the H. Here the H is mirror image to the H, like far. OH and OH are near to one another. CH2OH with CH2OH, so they are mirror images. Non-superimposable, it means they have difference. If we just cut this thing and just copy paste into this thing, we will have the OH here is on the left, ولكن the H here is on the left, and no matter how much we moved it and oriented it, it will never come exactly the same. Because of that, we call it as the non-superimposable. And this is everything we have in this slide. I just want to make sure, is everything crystal clear أو لأ? Okay. All good? ممتاز. تمام. Perfect. Sure. Okay, so I have a question. Okay, so in regard to the enantiomers, yeah, basically in the enantiomers, we don't look at the difference in the carbon. We can actually have difference in one carbon, maybe a couple of carbons. Like we don't look at the enantiomers and the difference of the carbons as much as we look at it of being as mirror image and non-superimposable. Like this is the most important thing into the enantiomers. And usually, if the doctor wants to point that the answer is diastereoisomers or epimers, they usually get like a sequence of carbon molecules. For example, 4 or 5 carbon molecules, عشان to know is there difference in the for example, in one carbon or two carbons or three carbon, in one carbon, two carbon, or لا. in compared to the enantiomer. For example, enantiomer, he can only get you, for example, a 3-sequence carbon, and you can only see one carbon, which is like attached in this side to OH and H. So you won't really know, is this, for example, epimer, ولا مو epimer, ولا like this. But usually, if the answer is going to be epimer, ولا distereoisomers, they should get a sequence of carbon molecules عشان to differentiate this. It's not to mix up with the enantiomer and everything. So is it clear? Good. Exactly. In the enantiomer, you can definitely know if it's D or L. As we said, D is the right side. معناها the first carbon molecule that comes after the aldehyde, or in other words, we can say the second carbon molecule. Once the OH is on the right side, it's D. Here, in this case, once the OH is on the left side, it's L. يعني this is the difference between the D and the L. Is it clear? And basically, we can say DL in whatever thing, enantiomer, diastereoisomer. It doesn't really matter what type of isomer it is, as long as it's basically a carbohydrate molecule. تمام? Clear? Any questions? All clear, ممتاز. تمام, as I said, like this line is quite important. Take your time into it. If you want to repeat the video, repeat it. Okay, so I have a question. Okay, and the epimers type. So basically, this is an epimer. Yes, tamam. So basically, as we said, epimer, the most important thing about epimers is that we have one different carbon molecule. مثلا, as we said in here, all of them are exactly the same. This H is on the right, H on the right, except in this one, this is the most important thing to know about the epimer. And we said that we have the exact chemical formula. D means it's on the right side, L means it's on the left side. Tamam, tamam. So basically, I get your question. Basically, here D means it's on the right side. And in right to Manos, it should be on the right side, but it's on the left side. Consider this as an exception. This is how it looks, but generally speaking, if the doctor wanted to ask you about D and L, D is always OH is on the right side, and L is always on the left side. Tamam? Just don't bother yourself about this Manos thing. Clear? Okay, about the mannose, I know here it's written as D. Like, see, the most important thing about epimer is just to know we have one, epi means one, we have only one carbon molecule, there's difference. This is the most important thing to know. L, D, and everything, just know the concept. D is on the OH is on the right side of the, like the first carbon that comes after the C double bond OH, and my L is on the left side. In regard to this mannose thing, I know here it's written as D. It should be on the right side, the OH. I can consider this as an exception. Tamam? Exactly. We look at this carbon, the first carbon after the C double bond OH. تمام? The C double bond OH, okay? So this carbon has the OH on the right, this OH is on the right, خلاص, it's D. For example, if we have it on the left, as we can see, this C, the OH is on the left, it's L. Okay? Clear? Any more questions? Ask as much as you guys want, عادي. خلاص, all clear? تمام. What about diastereoisomers? Okay. صح, احنا we said R is D معناها is on the right side, وهنا D معناها is on the right side. For example, of glucose, the D معناها OH is on the right side. If you found for anything D, the OH is not on the right side, consider this as an exception and never bother yourself about it. If the doctor wants to get you a question, it will be very crystal clear that D, the OH is on the right side, and the after the C double bond OH, ولكن L is going to be on the left side. خلاص, if you have this space, it's clear. Never mind the exceptions. تمام? Clear? As long as you know that what's stereoisomer, what's enantiomer, what's like this is the most important thing. Okay? تمام, ممتاز. Any other questions? Perfect. تمام. Okay, let's move on. خلاص, this slide is the bulk of the whole lecture and the most important thing. Okay, so I can't emphasize it more. طيب, so we have no questions till now, صح? ممتاز. Okay, let's move on. So let's get to know about the concept of the structural stereoisomers. So we have in here the compound with the same chemical formula. They have different arrangement, as we said. This is basically the atomic element that are called isomers, as we already explained previously. We have the stereoisomers. They have the same exact chemical formula, ولكن they have in regard to the 3D dimension into the space, they are quite different. بالنسبة للenantiomers, as we said, they are mirror images to one another. and they are non-superimposable. معناها if we put them on top of each other, they will always be different and they won't be exactly the same. And as we said, we have the D and the L. D is basically the right side. The OH after the C double bond OH is on the right side. And L, the OH is basically on the left side. And in regard to the stereoisomers, they have different configuration around one carbon atom. And if we have only one carbon atom difference, it's called as the epimer. Epi means one. So this is just explaining whatever we had before. So if before you understood everything in this previous slide, this means that inshallah everything else is going to be way easier, and we'll just repeat each and every concept عشان to solidify it inshallah. Okay, so we have in this slide, it basically says that the predominant carbohydrate encountered in the body, they basically are structurally related. And for example, you have this word aldotriose. Let's say that you don't know this word. Once you see anything that ends with OSE, خلاص you know it's sugar. And trio is basically three, so three carbon atoms of sugar. And aldo is aldehyde, so it have a C double bond O attached to an H. So خلاص you already orient yourself what's the meaning of aldotriose, تمام? So we have aldotriose with glyceraldehyde and ketotriose with dihydroxyacetone. Usually acetone comes with keto or ketose. Now, for example, we have ketotriose. We know O's is sugar, tri is 3. This the difference between the ketotriose and the aldotriose is that here we have aldehyde group, which is the C double bond attached with O and then H. أما بالنسبة للketotriose, it has C double bond O, it's attached with another C atom from either sides. Okay, so now we already know what's aldotriose and what is ketotriose. ممتاز. Now we have here all carbohydrate contains at least one asymmetrical chiral carbon. For example, we have here orientation of the hydroxyl group is quite important because it tells us actually is it the D or is it the L? تمام. Now we have with a few exceptions, we have this carbohydrate that are physiological significance and the configuration. We said D is the right side and the L is the left side. And enantiomers are basically mirror images that are non-superimposable. Okay, so I just want to clarify something. Okay. Okay. So I guess I said something wrong. Okay, so guys, I want to tell you about the D and the L. We said D, that means the OH is on the right side, ولكن L, the OH is on the left side. It's actually not the first carbon after the C double bond OH. It's actually the last carbon after it's like the second last carbon after the C double bond OH. For example, I'm sorry, I'm mistaken in this thing, and I just want to clarify it again. So basically, we said in this thing, for example, we have 3 carbon molecules. If the OH in this side, the second last carbon, not the last one, which is this one, the second last, for example, like this one, was on the right side, it means it's D, as we already said. If it's L, which is the left side, if the OH is basically on the left side, if the OH is on the left side, that means it's L. So we already know this thing. It's the second last carbon. Same goes into here. We said in regard to this thing, the D, it means it's on the right side. D is on the right side. D is on the right side, and anything L is on the left side. So for example, we have the D in this thing. The OH should be basically on the right side. صح صح. أنا by mistake I said the first carbon, the second carbon OH after the C double bond O should be here on the right side. I'm sorry, I meant the second last carbon, not the second carbon, the second last carbon should be on the right side. For example, we have the OH in here is on the right side, that means it's D. Same goes onto here, the second last. It's not the second carbon, no, it's the second last carbon. If it's on the OH on the right side, that means it's gonna be D. Same goes into here, D, glucose, that means that the OH should be on the right side. It's the second last carbon, which is this one, should be on the right side. Same goes into here, D, that means the OH should be on the right side, the second last carbon, not this carbon, not this carbon. لا, it's not the second carbon, it's the second last. يعني قبل الأخير, the carbon that's before the last one should be on the right side to be D. I just want to make sure, is this clear أو لأ? Okay, perfect. ممتاز. Clear guys? خلاص? ممتاز. It's not the second, it's the second last. Okay, so here the second last carbon, the second last carbon, same goes into here and same goes into here, the second last carbon. If it's the OH on the right side, it's D. If it's on the left side, that means it's L. Clear? Okay, ممتاز. Perfect. Okay, moving on. Okay, so basically same goes into here. We have the D. and the L. D is basically glucose on the right side. D glucose means the OH is on the right side, L is on the left. So we have this is on the left, this is on the right. The second last carbon. So this is the last carbon we have. This is the second last before it. It's on the right. خلاص D. It's on the left. It's L. تمام. Crystal clear? Okay, ممتاز. Perfect. تمام. Moving into the enantiomers is just basically repeating all the concepts which we said before. So basically they are stereoisomers that are non-superimposable no matter how much they are on top of each other. They won't exactly be the same, and they are mirror images. We have in here, as we said, the D and the L. D is basically the OH on the right side. L is the OH is on the left side of the second last carbon. And the second carbon, second last, no, before the last one, is on the right side, D and L. And basically D and L can be written before any type of the carbohydrate. We have, for example, here the D glyceraldehyde. For example, D is on the right side, the OH is on the right side, L, the OH is on the left side, and we have over here the glyceraldehyde. Glycer is something related to glucose. Aldehyde, because we have the aldehyde group, which is C double bond O attached to the H, or CHO in other words. Same goes in here. Glyceraldehyde, you have like something related to glucose, and the aldehyde group. تمام. And it's designated as D or L. As we said, D is right, L is left. تمام. ممتاز. Same goes to the distereoisomers. Distereoisomers are basically isomers or stereoisomers that are not mirror images, and they are just considered to be water soluble. And in regard to the epimers are basically epi, معناها they are only different in one carbon atom. That means all of them are going to be exactly mirror images of one another, except in one carbon atom they will be different. And yeah, so basically we have in here, just say the difference between the galactose and the mannose. Galactose is basically an epimer of glucose, for example. This is number one. For example, we can see here, this is the galactose, and this is the glucose. Now, this galactose is epimer. As we said, epimer معناها all of the Fisher configuration is exactly going to be for both the same, except only in one carbon atom. So we have over here the galactose, and this is the glucose. So they are epimer to one another. This is also quite important, as usually in the exam they can get two figures, and they will ask you, is it epimer? Is it, for example, something else? So يعني يعني practice it. بس إن شاء الله بإذن الله it will be like so easy. And we have in here the mannose is epimer of glucose. So we have this is the mannose. It's epimer of the glucose. Type. So basically the difference between both of them, for example, this one and this one is only in carbon number 4. We count from top to bottom. So this is number 1, 2, 3, 4, 5, 6. Why do we count from the top? Because it has the hydroxyl group, which is the double bond. Usually, if we want to count, we count from the double bond, and then like we start from the carbon with the double bond, and then move all the way downwards. So we count like 1, 2, 3, 4, 5, 6. And in carbon number 4, both of them are basically different, because here the OH is on the left side, but I can hear the H is on the left side. Compared to other carbons, they are exactly the same. For example, H on the left, OH left. Here the H on the left, so on and so forth. So both the galactose and the glucose are basically epimers. Now, comparing the glucose to the mannose, they are exactly the same thing, except in carbon number 2, they are basically different. Why? Because here we have the H is on the left side, but in the OH in this case is on the left side compared to everything else. OH is the left, this is left, H is left, and H is the left. So glucose and mannose are epimers to one another. Clear? Perfect. Okay. ممتاز. Everyone clear, صح? Perfect. ممتاز. Okay, moving on. So as we said in here, we have the Fisher, we have the Haworth, and we have the Kayar or the shear style diagram. Basically, Fisher it looks like this, Haworth is like this, and the Kayar or the shear, it looks basically like this thing. تمام. So we have three different types of how the carbohydrate molecule looks like. ولكن at the end, this is exactly the same as this structure, and this structure is the exact same thing as this structure. Okay, so they're the same, but they look different into the space by having different configuration, either Fisher, Haworth, or shear style configurations or diagram. Okay, here we're repeating the exact same thing. We have over here the carbon molecules. If we come to fold it, it will be like this. And this is the final ring structure of how it's basically folded. As we said, this is the anomeric carbon, or it's basically carbon number 1 atom. Tamam. So if I want to count from a ring, I usually count from the anomeric carbon, which is carbon number 1. Why do we call it? Why do we usually like count from number 1 carbon? Which is in this case this one, because it has the double bond O, and it basically has the aldehyde group. So we usually count from number 1 in here all the way onwards. So this is number 1, 2, 3, 4, 5, and this is number 6. Tamam. So we have this 6 carbon structure carbohydrate, which is glucose. Why do we call it D? Because the hydroxyl group or the OH group is basically on the right side. As we said, if it's a ring structure, it's quite difficult to know if it's on the right side or left side, but usually if it's like this structure, it's way easier to be knowing if it's on the right side or on the left side. Now, in regards to the ring structure compared to this fissure structure, the only like most common difference between them is that if you want to call it as alpha or beta, it should basically be in a ring structure compared to this thing. And if it's like a fissure structure, you can it's easy to say if it's D glucose or L glucose. ولكن if it's in a ring structure, it's easier basically to be knowing if it's alpha or beta. Now, as we said before, if it's alpha, that means the OH and the CH2OH are basically opposite to one another. How is it opposite? This OH is pointing downward compared to this CH2OH is pointing upward. So basically, if there's a difference between, if they're the opposite, that means it is alpha. Just remember alpha. It's like O is opposite. So they are different from one another. That means it's alpha. If OH and the CH2OH are both pointing above or both pointing to the same direction, that means it is beta. So this is the difference between the alpha and the beta. See, like all other slides are basically just reviewing the previous concept. Tamam. So, okay, perfect. So as we said in here, this is the anomeric carbon, and this is how it basically looks like. This is the fissure or the linear skeleton, and this is the ring-like skeleton of a 6 carbon molecules. Clear? Perfect. تمام ممتاز. Okay, so now let's get to know more about the pentose and the hexose. As we said, pentose is basically a 5 carbon sugar compared to the hexose, which is a 6 carbon sugar. Anything that ends with OSE is a sugar. Anything that ends with the OSE is a sugar. We already know what's the anomeric carbon. Once the ring closes, this carbon, the first carbon, is called as the anomeric carbon, which is the new chiral center carbon. And we already know the difference between the alpha and the beta. As I said, alpha is basically opposite side. Beta is the same side. If both of them are called the same side, or beta, we call it as cis. If they are in the opposite side, alpha, we call it as the trans. So same side, cis, opposite side is trans. Perfect. This is the exact same thing. We have in here the linear skeleton. We have in here, it turned out to the ring-like structure. This thing is basically called as the how many carbons we have. You can count it 1, 2, 3, 4, 5, 6. We have 6 carbons, and usually we count from top to bottom because the aldehyde or the C double bond O is found here, and usually this is carbon number 1. If this group is found anywhere else, not in the above, we're gonna call from this exact point. Yeah, and it's not because it's the first, we're gonna count it from the first. La. We see where is this aldehyde group, the C double bond O, and then we start counting from it. Usually it's available at the first carbon. So we have in here 1, 2, 3, 4, 5, 6. And then if we want to all the way close it into a three-dimensional shaped ring structure, it's going to all the way fold into this thing. And then it's going to end up being like this. As we said, this is the anomeric carbon, and it's here called as beta. So why is it called as beta? Because we have the OH and the CH2OH, both of them are pointing upward. This is pointing upward, and the OH here is also pointing upward. So both of them are the exact same direction. It means it's beta. Compared to this thing, it's alpha. Why? Because the OH in here is pointing downward. Compared to the CH2OH in here, it's pointing upward. So this is down, this is up. Both of them are opposite. That means it's alpha, or in other words, it's trans. Tamam? Okay. So moving on, the exact same thing. We have over here the pyran or the pyranose sugar, and we have over here the furanose sugar. Just remember, how can you remember like pyran is how many sugar? If you just flip the P letter, just flip it, it's gonna look like a 6. So basically this is a 6 carbon the 6-carbon sugar molecule compared to the furan. Just remember, furan is like having a furan, yeah, like an oven. So this furan take 5, for example, cupcakes. So furan, like a furan, is basically 5 carbons, and a pyran is basically P. If you just flip the letter P, it will turn to 6. So basically, it's a 6-carbon molecule. Just easy way to remember how many carbon molecule each carbohydrate has. For example, in this case, we have alpha. Why is it alpha? Alpha and beta is also quite important, like they usually like to ask about it. Alpha is opposite side. That means the OH and the H, and this HO or HCH2 is up. So this is down. This is up. That means it's alpha compared to this thing in here. The it's basically also alpha. Why? Because this OH is down and this CH2OH is up. That means both of them are opposite direction. It's going to be called as the alpha. Okay, so is everything crystal clear till now? Tamam, perfect. Okay, moving on. Okay, so we have over here the carbohydrate, the monosaccharide, oligo, and the polysaccharide. As in the polysaccharide, poly means many, so we have a lot of carbon molecules, and they are generally containing hundreds of the monosaccharide. What does monosaccharide mean? Mono معناها it's only one simple one simple sugar, and it's basically it can't be more simpler. يعني monosaccharide is this is the basic form or as a basic let's say structure of sugar, and it can't actually be like this is the least amount of sugar. We can't have less than mono. So like mono is the first thing. Di means we have two carbohydrate molecules, for example sucrose, fructose, and sucrose, for example, is made of two things, which are what, the glucose and the fructose. So glucose and fructose. Glucose is monosaccharide, fructose is monosaccharide. So glucose and fructose, mono and monosaccharide, turns out to be the sucrose, which is the disaccharide. Same goes to the oligo, generally speaking, means few. So basically, few number of saccharide or few number of sugars. In this case, it can be 2 to 10 monosaccharide or 2 to 10 carbon molecules, monosaccharide units. And in regard to the polysaccharide, we have actually a lot of monosaccharides, a lot of carbon molecules. Okay, so we have here a quick question: what are monosaccharides? What do you guys think is the answer? So simple. Perfect. Exactly. Mumtaz. Yeah, it's actually B. As we said, mono means one, so it's only a single sugar molecule. Excellent, mashallah. Okay, so let's repeat what's monosaccharide. As we said, monosaccharide is only one carbon molecule, so they are commonly found in human. And the most common type of carbohydrate is basically glucose, as being the most common monosaccharide, and they are basically physiologically important in the human body. As we know, we need like carbohydrates in general. So moving on, okay, so basically this thing exactly explains the thing we had before. We have the aldose and the ketose. We said O's ends with sugar. O's is sugar. Aldose because we have the carboxyl group, C double bond O with H. And here we have the C double bond O is attached to a C carbon atom. So this is the difference between the aldose and the ketose. Triose is basically 3. Tetrose, tetra is 4. As we said, O's is sugar, or anything that ends with O's is sugar. Pent is 5, 6, and 7. Okay, so we have in here just the name of the, as we can see here, triose is C3, C4, C5, C6, and C7. We have in here, for example, an example of triose. In this case, for example, the glyceraldehyde. Here we have the dihydroxyacetone. As we said, aldehyde, aldose, acetone. Acetone is mostly to the ketone or the ketose. In regard to the tetrose, we have in here this example, same goes to the pentose, this one and this one, same goes to the hexose. We have glucose, fructose, like so on and so forth. Just example of the names of the different type of carbon molecules, 3, 4, 5. Here we have also sugar. How will we know that those are sugars? Just look at the end. It ends with O's, 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 O's. خلاص. That means it's definitely a sugar. D configuration معناها إن the OH is found on the right. L معناها إنه the OH is available in the left, and here it basically explains the source and the importance of the ribose and the ribulose. Basically, ribose is a nucleic acid, and a ribulose is a metabolic intermediate, and just states where basically they are available and how they are used and their clinical importance. difference between both the ribose and the ribulose compared to the rest, which are not as important as or not as commonly used as the ribose and the ribulose. تمام? Okay, so moving on. Okay, here we have just extra information about glucose, fructose, galactose, and the mannose. As you know, glucose is the most common type of the monosaccharide, and it's available in the fruit, it's available in the beet sugar, and it's a basic and the main metabolic fuel. And usually, once we measure the glucose level in the blood, we actually check for the glucose levels, and if it's high, that means the patient either ate sugar before, or the patient is diabetic and isn't, for example, getting their diabetic medications. And this is the clinical significance of the glucose. As we said, it's used. It's usually high in diabetic patients or patients who have diabetes mellitus. And yeah, so either the patient isn't compliant to the medication, either the patient should increase the dose of the medication or like whatsoever. In regard to the fructose is basically a fruit juice, and it's basically a sugar of the fruits. It's also available in the honey and many other different things. And okay, so for example, patients can have actually hereditary fructose. And in regard to the galactose, we have galactose and the mannose. Galactose hydrolyzed into the lactose compared to the mannose, which hydrolyzed into the plant manna and gums. And basically, these are, to be honest, it's not really as important as the slide of the isomers, of the stereoisomers, of everything we explained. That slide is like the most important thing, and it has like almost all concepts. Like compared to those slides are like more of extra of knowledge. Okay, as we said in regard to the oligosaccharides, saccharide is sugar, oligo is a few. So we have a few number of sugar or carbon molecules. It basically contain 2 to 10 monosaccharides, and they are basically, many of them are non-digestible or indigestible, and by our body enzymes. And oligosaccharide can increase the viscosity of the gut. This is just extra knowledge. So as we said in here, in regard to the oligosaccharide, we have this table basically is this table is quite important in regard to this thing, which I'll explain in the coming slides. So basically, we have in the sucrose, in the lactose, in the alpha trehalose, and in the maltose, and in the silobiose. It basically just explains where the molecules basically where the ring structure are bonded to one another, and this just explains the bond. This bond is quite important, and they usually get it in the exam, so I'll explain inshallah more about it in the next slide. And this just says where the sucrose is available in fruits, lactose, milk, and so on and so forth. Okay, now this slide is the second most important slide in this lecture. Tamam, tamam. The first slide was the one with lots of figures that we explained it, and this is the second most important slide. So we have in here 3 examples, the first, the second, and the third. As we said, to know the D and the L configuration is quite difficult in those structures, and we mainly use those structures to know is it alpha or beta. As we said, alpha is opposite the OH, and the CH2OH is opposite alpha. If they are both the same direction, it's beta. This is clear. So we usually use the ring structure to know is it alpha or beta more than to know is it D or L. D or L if it's a linear Fisher-like structure. تمام؟ Clear لحد الآن? Is everything clear till now? ممتاز, perfect. طيب, now we have in here 2 ring structure. This is the first ring structure, and this is the second ring structure, or in other words, the first glucose, the first carbohydrate structure, and the second. Same goes to here, and same goes to here. Now I'll focus on the first one. Now we have this left structure and this right structure. We agree that both of them are carbohydrate structures. Alhamdulillah. We also agree that this is carbon number 1. And also we agree that this is also carbon number 1 in here, which is the anomeric carbon. Alhamdulillah, this is clear. Now we have those 2 structures should bind together, and they will bind together through a bond. Tamam, tamam. For example, we have in this case, they will bind in this side, they will bind in this side, they will bind in this side, because it's easier than this is going to bind with this, or like common sense, this to this, like they are close to one another, so they will bind together. Now, this name is basically the name of this bond. Usually how they get in the exam this question, they usually, for example, get a figure and they will tell you what's the name of this bond. Is it this one? They just give you a couple of choices. And for example, they will say, is it this one? Is it this one? And like other things. So this is the correct answer. Why? Let's look at this thing. So basically we will look at the at the left-sided structure first and then at the right-sided structure. Now I'll start off explaining with this structure. This structure has the OH above and the CH2OH is above, so both of them are the same direction. Is it beta or alpha? No need at all to know the ring structure, no need at all to memorize anything. Just know the concept. If you know the concept, no matter whatever structure or figure the doctor got you, inshallah you're gonna get to know it. As long as you understand the concept, like this is the most important thing. Tamam? طيب, now my question was, we have the OH and here is up, and as you can see, basically this anomeric carbon, the H is down, so the OH, this is the O, and they removed the H because we made a bond with it. So basically the OH is above, and here the CH2OH is above. So this is above and this is above. Both of them are same direction. Is it beta or alpha? ممتاز, beta. Excellent. Bravo. Basically it is a beta. So because of that we have here beta. Now we know what is this beta. And we have in here number 1 because we have carbon number 1 is basically is the one that's making bond with this carbon number 4. As we said, this is 1, 2, 3, and 4. So this is carbon number 4. So 1, so it's basically beta because this OH is above with the CH2OH, and also here this OH is above with this CH2OH. So both of them are above, having the OH is above with the CH2OH. So up and up are same direction. صح ولا لأ? If both of them are same direction, معناها it's beta. So it's beta. And the bond is 1, this bond is 1, going all the way to this bond, which is number 4. So 1 to 4. So now we understood what is this beta 1 to 4. Is it clear to everyone ولا لسه? Perfect, clear. Okay, ممتاز, it's clear, ممتاز. That's great. Now we have in here, this is the gal, is basically galactose, and GLC is basically glucose. So it just says that this molecule is galactose. Compared to this molecule, it's glucose. What we care about the most is this thing, beta 1 to 4. Both of them are up, OH with CH2OH in both figures, so it's beta, bond number 1 with bond number 4. خلاص, alhamdulillah, the first example is clear. Let's practice more and move to the second example. Now we have, they will get just a figure like this. They will tell you what is it. Is it, for example, this thing, or is it something else? So you will first get oriented to the ring structure. مثلا, we have this as the anomeric carbon, so it's carbon number 1. We're gonna count 1, 2, 3, 4, 5, 6. Same goes into the structure. In the structure, we have the it's basically, as you can see here, this is a 6 carbon structure compared to this thing, which is a also 6 carbon structure. ولكن it's it looks different. For example, we can see here it's like this, here like it's a triangular shape. Compared to this thing, it's not the same thing. So we should the numbering is basically changed. Usually we will number it from the C double bond O. صح ولا لأ? صح. So in this case, this bond is basically available here. So we'll count it as 1, 2, 3, 4, 5, and 6. تمام تمام. Now we have in here, I'll start off explaining this thing in here. Basically, this answer is exactly the same as this thing. They just flipped it. For example, هنا glucose alpha 1 and هنا glucose alpha 1. يعني it's basically كأنه left to right, right to left. So I'll start off explaining with this thing عشان it's easier to our figure. So we have this as a glucose structure. So they wrote glucose and then they wrote here alpha. Why is it alpha? Because in this carbon we have the OH here is pointing downward. ولكن the CH2OH here is pointing upward. So down and up are opposite. That means it's alpha. And alpha and which number of carbon? And carbon number 1, which is this carbon, the anomeric carbon. خلاص. So now we agree it's a glucose alpha 1. Now here they told us it's beta. Why it's beta? Because here we have the OH, and we can see here, this is the bond that's bonded with the other structure. So this OH is down compared to this CH2OH is also down. That means this is down and this is down. Both of them are the same side, so it's beta. What's the number of carbon it is? It's basically carbon number 2. So we have in here glucose alpha 1. This is glucose. This is alpha 1, carbon number 1. Is paired all the way with the second carbon, and it's beta because this is pointing downward and this is pointing downward, the CH2OH. So this is down, that means it's beta, and it's basically a fructose sugar molecule. So this is how we name it. In other words, this is exactly the same to it, بس يعني they just flipped it. مثلا هنا they wrote glucose alpha 1, which is here glucose alpha 1, fructose 2 beta, it's here fructose 2 beta. Is it clear, or do you have any question? Excellent, clear, ممتاز. Perfect. ممتاز. Perfect. Okay, so we have in this structure, it's exactly the same thing. This is our third example. We have, this is the anomeric carbon. So we're going to count as 1, 2, 3, 4, 5, 6. And we have in here the OH is pointing downward compared to the CH2OH is upward. So downward and upward, they are opposite to one another. That means it's alpha. Compared to this thing in here, the OH is basically up. Compared to the CH2OH, it's basically down, as we can see in here. So this is down, this is up, they are opposite to one another. That means it's also alpha. So this is glucose alpha 1 all the way paired with glucose alpha 1. This is the name of this bond. Usually how they get this, and this bond, it's basically called as the glycosidic bond. How they get this question in the exam is basically they get just a figure like this thing, and they will tell you what's the name of this bond, and they basically give you a choices, and in this case, this is basically the correct answer. Just tell me, is this important? Is this understood? Did you guys understand, or do you want me to repeat any point again? Because this slide is quite important. Clear, everyone? Okay, perfect. طبعا clear, ممتاز. Okay, moving on. So basically, there are no any questions. Any questions? No, tamam, perfect. Okay, so now let's get to know more about the polysaccharide. As I said, polysaccharide is basically many saccharides. This is the meaning of poly. Poly means many. Saccharide is basically a sugar. So we have a couple of sugars or many sugars. Usually it's considered as more than 10 monosaccharides because we said oligo is 2 to 10 sugar molecules. Polysaccharide is going to be more than 10. So this is the meaning of the polysaccharide. Now in regard to the polysaccharide, we have two things. Either it's going to be plant-derived saccharide or animal-derived saccharide. If it was plant-derived saccharide, we have two types, the amylose and the aminopectin, compared to the animal, which is the glycogen. Tamam. Now, what's the difference between them? In regard to those bonds, they are quite important to like memorize. For example, in regard to the amylose, it's basically linear molecule. Aminopectin is branched, so it's linear, and usually the bond is at alpha 1,4. It's important to memorize that amylose is linear and has a bond at alpha 1,4. Compared to the amylopectin, it's basically branched and it has a bond at the alpha 1,4. It's all the way bonded with alpha 1,6. So this bond and this bond, it's quite important to know that those two bonds are basically for the amylopectin compared to the amylose, which is only one bond at alpha 1,4. Now, in regard to the glycogen, it's basically branched at alpha 1,4, alpha 1,6 bond. So basically, like to know the name of the bonds. For example, amylose has bond of like this, amylopectin and glycogen has bond of like this and like this. Those are quite important, and it's also quite a high yield question. So let's get to know more about and understand them. As we said in here, to talk more about the amylose and amylopectin, we said no. Amylose is basically a linear structure compared to the amylopectin. It's basically branched. So as we can see here, any structure that's linear like this is amylose, and a structure that is branched is basically amylopectin. And we get to know the starch. Starch is basically it's the major and the most important form of the stored carbohydrate in the plants, and it basically either can be amylose or can be amylopectin. Amylose is not branched compared to the amylopectin, it's a branched, okay? So moving on, we have the amylose. As we said, amylose is not branched. We can see, for example, like this three-dimensional structure, it's not branched to any other thing compared to the amylopectin, which is branched. So as we know, amylose is basically a linear polysaccharide, and it basically has the hydrogen bond to stabilize this 3D helix structure. And we have basically the amylose is used to slow the digestion. And example of it is basically lentil, and it's a human being like human being. And for example, we have the legumes, we have the long grain rice. All of this are considered as amylose, and they have low amylopectin starch. That means if they are amylose, they are all 3D dimensional, that are linear in structure, and they are not branched. This is the most important thing about amylose. Comparing it to amylopectin, as we said, it's going to be branched. For example, we have in this structure, you can see there is a branch, it's branching, like it's connecting to another thing. Same goes to this structure, it's connecting to another thing. Here we have in this bond, it's basically alpha 1,4 glycosidic bond. Why is it alpha? Because the CH2OH is on the opposite side of the OH. They are on the same side. This is why it's alpha. And 1,4 because the bond is basically happening between carbon number 1 and carbon number 4, and the name of the bond is basically a glycosidic bond. So this is more about the amylopectin compared to the amylose. Amylose, they can rapidly be removed, and glucose monomers and oligomers, and so this is the difference between the amylase and the amylose and the amylopectin. And here it just says about the enzyme amylase. ACE is basically an enzyme. It just rapidly removes glucose monomers and oligosaccharides in the rapid blood sugar and in short period of satiety. Okay. So moving on to the glycogen, as we said, glycogen is the major form of stored carbohydrate in animals, and usually the carbohydrate or the sugar is stored in our liver as glycogen store. Whenever our body needs this store, it just sends out the sugar molecules for the body, or ATP molecules, so that our body is going to be taking the power. And as we said, the majority of the glucose monomers, they are actually linked by alpha 1,4 bond or alpha 1,4 linkage. And glycogen also has branches, as we can see in here. It's basically branched. It can be branched anywhere, and it's basically once it's branched, or once it forms a bond with other structure, it's usually called as the glycosidic bond. تمام. So moving on, we have a variety of complex carbohydrate containing complex compounds that exist both structurally and functionally. We have basically the glycoprotein, the glycolipid. What's the meaning of glycoprotein? Glyco is basically sugar, protein is protein, so glucose and protein, glucose and lipid, glycolipid. We have the glycosaminoglycan, we have the proteoglycan. So just whenever you see a long word, it's going to be simple if you guys just divide the words. And yeah, so basically we have in here the membrane associated carbohydrate. As we said, carbohydrates can be available either as proteoglycan, glycoprotein. It can be available as energy, as cell signaling. So it can actually be used not only as a source of power, but as many other things, and can help in cell signaling and many other functions in the body. As we said, we have the glycoprotein and the proteoglycan. So glycoprotein is between the protein and the carbohydrate, and the proteoglycan, the carbohydrate, and the protein. تمام. So yeah. Moving on, as we said, we're going to explain more about the difference between the proteoglycan and the glycoprotein. Proteoglycan, that means we have the carbohydrate more than the protein, compared to the glycoprotein, which is the protein more than the carbohydrate. In regard to this thing, the carbohydrate portion is always a heteropolysaccharide, and the carbohydrate is long and linear. Those are the two examples of the proteoglycan, compared to the glycoprotein, where basically the carbohydrate portion is either monosaccharide or oligosaccharide because it's less. Just remember, if I say glycoprotein, that means the second word, which is the protein, it's more. If I said proteoglycan, that means the glycan or the glucose or the carbohydrate is more. This is just a basically simple way to memorize the difference between them. So proteoglycan, خلاص, the second word, which is the glycan, is more. Glucose is more. Glycoprotein, that means the protein is more. تمام. This is just an easy way to know the difference between them. Now, in regard to the glucose, the carbohydrate, we said it has a couple of functions. One of which is basically it should basically be having a transporting from one place to another. For example, if it's available in the blood, in the heart, in the liver, wherever it was, it should be like moving from one place to another. And basically it's available in the whole body. So we have, in regard to the transporters, like more talking more about the carbohydrate or the glucose transporters, we basically have GLUT1, 2, 3, 4, and 5, and each of them are basically available in different places. For example, GLUT1 is available in the blood, in the BBB, which is the blood-brain barrier, in the heart, compared to the GLUT2, which is the liver, pancreas, small intestine. Dr. Jada like to focus more on GLUT2, which is available in the liver, pancreas, and small intestine, because as we know, we talked more about the glycogen storage and glucose, so it's mostly available in the liver compared to any other places. As we know, the most important energy for the brain is basically glucose or sugar in general. We have also GLUT4, it's available in the skeletal muscle, adipose tissue, heart, and the GLUT5 is available basically in the intestine, compared to the second glucose transporters, which are basically the SGLT. We basically have two types, 1 and 2. SGLT stands for the sodium dependent glucose transporter, or the sodium and the glucose 1 transporter with the sodium and glucose 2 transporter, and they are basically available also in different places with different functions. But the most important thing to focus on is GLUT2, which is basically available in the liver, pancreas, and the small intestine. The most important thing is the liver. Tamam? Okay, so moving on. Okay, so we're gonna talk most about the GLUT2. As we said, the doctors prefer talking most about GLUT2 because it's available in the liver. If we have any deficiency in GLUT2, we have a syndrome which is called as the Fanconi-Bickel syndrome, and this leads to the accumulation of, for example, the glucose is available in the liver and doesn't allow it to be all the way going outside to the blood and to be like going anywhere else in the brain, in the body. That means we're gonna have very high accumulation of glucose and glucose storage, which is the glycogen in the liver, which is somehow going to lead to many complications and problems in the body, and this is known as the Fanconi-Bickel syndrome. And we have basically passive transport happens between glucose, galactose across the GLUT2 transported in the basolateral of the membrane. We have also, it basically can not only include the liver but also the pancreas, the renal tubule or the kidney, and the intestinal epithelium. And just says here, if there's an inadequate export of the glucose, it leads to very, very high accumulation of the glucose intracellularly in the liver, and in this way there is an inhibition of the glycogen storage to be all the way degraded and send us the glucose away from the liver throughout the body. Now, in regard to this slide, it just basically says if we have, for example, very, very, very high levels of glucose in the body, or in other words, in the blood, what is it going to happen? We're going to have the glycosylation of the hemoglobin with the sugar molecule. So if we have a hemoglobin with sugar molecule, in a normal amount, it's normal. ولكن if it was very, very high, or in other words, if the patient was diabetic patient who has diabetes, they usually, if they weren't on their medication, like compliant to their medication, they will usually have, or if they aren't taking their medication, they will have very, very high amount of HbA1c, or the hemoglobin that's attached with the carbohydrate molecule. As we can see here, it's basically called as the glycated hemoglobin, or the hemoglobin that has the glucose is going to be significantly very high, and it will cause the age, or in other words, the advanced glycation end product. So basically, if the hemoglobin A1C is basically HB is hemoglobin A1C. A1C means glycosylated hemoglobin, or sugar in other words. So HB is hemoglobin, A1C is the sugar or the glucose. So if this is very high in the body, that means there is no quite good monitoring of glucose. Or for example, if you ate very sweety food or something, this is going to be significantly increased. So this is the meaning of HbA1c. If in any person is having constant high level of glucose in the blood, or high level of HbA1c, it will turn out to age or to advanced glycation end product, and this will definitely cause a lot of problems in the body. It will cause a lot of dysfunctioning of different organs in the body, like the retina, the cardiovascular disease, GI, cardiac problems. The kidney is going to affect all organs in the body will be affected with very high levels of glucose, and it will cause a problem in the cross-linking of the protein with problem in the transporting, problem in the like basically every single thing in the body, you just name it. Because of that, it's always better to control the intake and of the sugar. And yeah, so I guess this is it in this slide, and we're ending our lecture. So I hope you guys enjoyed. I hope you guys learned the most important, like the whole lecture is important, but the most important points to just focus on are basically the isomer, the slide of the isomers, the structures, and also the slide of the bonds, like what are the name of the bonds. These are the two most important high yielded questions that usually come in the exam. The rest of the lecture is also quite important, but like those are the most important things that the doctor likes to ask about it. We talked more about the D and the L. We said the D, OH is on the right side, L is on the left side. We talked about the alpha and the beta. Alpha is basically opposite. The OH and the CH2OH are basically opposite sides compared to the beta, which is basically the OH. And the CH2OH is basically on the same side. And we talked about the different structures that we can find, the different type of the isomers, and the glycosidic bond, how the glycosidic bond is basically available. We talked about the anomeric carbon. And yeah, so basically just focus mostly on these two things. And I hope you all the best of luck on all of your exams. If you guys need any help, please feel very approachable anytime. Just send me any question if you guys have any question, and inshallah, tawfiq and best of luck in your year. So I just want to know if you guys have any question or anything's unclear. Okay, everything crystal clear. Thank you so much. If you guys can kindly scan this code and just give your feedback on this session, and I would really appreciate it so much. And yes, thank you so much. Have a nice day. Thank you. Masalama.